Okay, so let's dive into some more of the science behind perinatal tissue and stem cells, and then we'll just do a little comparison for the next few minutes on the source of the stem cells. Because you've got adult stem cells in your adipose tissue, which is the fat that you may or may not have. You can also get it from your bone marrow. And one of the reasons when I was looking for treating my shoulder about 7 years ago for a rotator cuff tear, I looked at the different sources of stem cells, and I did not want to use my own stem cells because growing up on a farm in Idaho, I would move sprinkler pipes and I would have planes that would come down and crop dust the fields, and I would get drenched in Roundup. And so I ended up becoming pretty sick as a kid and in my early 20s, and struggling with some conditions that made me pretty vulnerable to the outside environment. So I looked at my stem cells, and they'd been exposed to everything I've been exposed to, just like your stem cells. Some of you guys are old, and now no offense, but as we age, like I mentioned in the previous module, our stem cell populations start to decline and our ability to heal starts to lessen. And so using this perinatal tissue, this this young source of cellular material, growth factors, cytokines, all the other the matrix that goes along with it, what I've discovered is that is how we can get the most reproducible results. And then as I dug into some of the research, I was able to find some of the comparisons between the umbilical cord stem cells and the bone marrow stem cells. So what they found is that in the umbilical cord blood, you're going to find two different types of stem cells: hematopoietic stem cells and mesenchymal, which also now are renamed by Arnold Kaplan medicinal signaling cells. And if you look at these, there's a higher immunological tolerance to the umbilical cord blood stem cells. So there's not what we see in this graft versus host response. We've also found that the stem cell survival is more efficient from umbilical cords than it is from bone marrow. So they're a little more robust, which makes sense because they're younger types of cells. If you look at the yields overall, if we were to compare how many stem cells per milliliter of tissue is in umbilical cord versus bone marrow or adipose, what you'll see is in bone marrow you're going to have anywhere from 1 to 317,000 stem cells per milliliter of tissue. In the adipose tissue, you're going to have anywhere from 4,700 to 1.5 million cells per milliliter of tissue. And then in the umbilical cord tissue, what they found is there's anywhere from 1 million to 4.7 million stem cells in a milliliter of umbilical cord. So kind of an interesting comparison as you look at that. And, we look at this and we say, well, there are stem cells that can be derived from the placenta, from the amniotic fluid, amniotic tissue, and the umbilical cords. And each one of these tissue reservoirs has a specific niche. So there's different types of properties that come from these stem cells. Not only is there the circulating growth and differentiating factors, and these factors determine the gene activation, and they also determine a functional reaction on stem cells, because stem cells, what we know about them is they behave one way in a petri dish, but when they get into the body, they behave in a completely different fashion. And so one of the reasons why we take all three compartments in the stem cell, in the perinatal tissue, we use the placental stem cells, the amnion stem cells, and the umbilical cord stem cells, is so that we can have the proper expression of healing based on what your body needs. And so that's one of the reasons why we use this differentiated piece. Umbilical cord stem cells, they've been shown to have plasticity. That means they can have the potential to change into other types of cells. Not only that, but even certain cells in the perinatal tissue is already differentiated down a specific lineage. So they may be like an endothelial cell, and those cells can differentiate and become a different type of cell in your body. The other thing is we found is they have homing capability. So they can travel to areas that have been damaged. For example, if we're injecting a knee for cartilage regrowth, they're going to go into that area and help start recovering some of that tissue damage. And then they have the engraftment capabilities where they can unite with other tissues. And this is something that we found very fascinating. There's a study that came out of China where they took 1,700 rats and they severed the sciatic nerve of these rats entirely in half. And they wanted to see if these rats would tolerate the immune stem cell, or from an immune perspective, the human umbilical cord stem cells that were injected into these rats. And in every case of the 1,700 rats, there wasn't a single adverse reaction. So they do engraft with other tissues very nicely. And so I think, this is what we've looked at from a safety perspective. We find it's very safe, very tolerable. And then also from an effectiveness level, these are younger stem cells with all the other factors combined. That's one of the reasons why we choose the umbilical cord stem cells or the perinatal tissue in general over like a bone marrow or adipose. However, those treatments are very powerful, very important, and certainly a better alternative to some of the surgical or drug outcomes and therapies that we've been recommended. So there's your science, and we'll jump in just a second and we'll talk a little bit more about cartilage.